Dear students, welcome to this MOOC. MOOC stands for Massive Open Online Course. In this MOOC, we shall be providing you a variety of multimedia enriched e-content, activities and assessments in a very learner friendly manner so that you are able to understand the various topics of this course very well. Let me now tell you briefly about the structure and main features of this MOOC. The entire MOOC has been divided into various weeks so that you can plan your learning in a very logical and systematic manner. It is advised that you must actively participate in the various components of this MOOC, that too regularly on a weekly basis. For every weekly schedule, you are first advised to watch 2 to 4 videos assigned to you per week. Then you must read the learning material provided as e-text and also go through other content in the Learn More section. After this, you are advised to carry out the various activities like taking part in the discussion forums, visiting the related web links which we have provided for you, attempting crosswords, etc., earmarked for each week. After this, to test your knowledge of what you have learned during this week, you must attempt the quiz in the assessment section for that particular week. I wish you all the best in this very interesting self-learning experience. Please remember, we shall always be there with you to provide you support for any problems you face. The MOOC coordinator along with junior faculty as tutor shall always be available to solve your queries which you can post in the discussion forums or you can send them to us through mail which shall be notified to you soon. We would be very happy to know your experience after the completion of this MOOC in the form of your feedback which will help us enhance the quality of this MOOC further. Dear students, after knowing the general MOOC structure, let us now try to understand the weekly structure of this course on Bioorganic and Biophysical Chemistry by the MOOC coordinator Dr. Nirmala.
The course Bioorganic and Biophysical Chemistry will be taught in a 15-week program with a break in the 8th week and 15th week for revision and assessment. The course content for each week has been organized in a systematic way so that from principles of biomolecular structure and purification, the biological function of enzymes to their applications are easily understood for a relative beginner. Emphasis has been made to the cellular location of various biomolecules which would therefore be relevant to their function. Techniques which are important to study biomolecular structure and location have been covered in the earlier weeks, whereas the kinetics and applications of enzymes are taught in the latter weeks. The end modules introduce you to the emerging areas of applications of bioinformatics and protein engineering to develop designer proteins with enhanced abilities to modulate biological functions and for applications in biotechnology. Today, science is interdisciplinary in nature and hence a study of basic sciences such as physics, chemistry, mathematics and biology at an undergraduate level have been put together at the postgraduate level in designing the present course. In the first 2 weeks, the structure of cells, their internal structures and methods of observing and studying them will be covered. An introduction to microscopy with their physical principles will be covered, and the exciting technique of FRET, which helps visualize molecules and cells in action will be covered in a video format. The various types of small molecules and macromolecules present in cells will be described with a module on DNA and protein structure. Modules covered in weeks 3 and 4 will cover protein structure, their specialized enzyme functions and techniques to extract and purify them from other cell components. Major characterization techniques of macromolecules such as mass spectrometry, CD and ORD will be studied. In weeks 5 and 6, the thermodynamics and kinetics of enzyme reactions will be described with a mathematical treatment to quantify the rates and efficiency of these biological catalysts. General enzyme mechanisms will also be discussed. Week 7 will contain modules to describe the mechanism of action of important enzymes such as Chymotrypsin, Lysozyme, Ribonuclease and Carboxypeptidase. After 7 weeks, there will be a break of 1 week wherein revision and assessment of the covered course will be undertaken with MCQs, true, false and match the following type questions. In week 9, the important role of vitamins and their coenzyme functions will be covered through 3 modules. Week 10 will cover modules in the various applications of enzymes in clinical research, food technology and therapeutics. These applications will also be studied in week 11 wherein the use of enzymes as drug targets will be covered. Modules covered in weeks 12 and 13 will deal with some fundamental properties of biomolecules such as chirality and their use as membrane active ionophores. Week 14 will cover the application of enzymes in biotechnology and modules will cover how designer proteins for human welfare can be created in the test tube. Week 15 will be reserved for a final revision and assessment of the course. After studying the course, you would be able to use the knowledge gained to understand a major area in biotechnology. I wish you a memorable journey through this course and hope you will appreciate the interdisciplinary nature of today's scientific research.